Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CUET entrance exam question paper for PG entrance. This is life science subject. It was conducted in the year 27th June 2023. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers been answered, you can go to my channel Omni Bioentrance. Go to playlist C for CUET. In that you will find these question papers being answered with explanation. So coming to the first question, they have given two statements. So you have to see whether both the statements are correct or one is correct or both are wrong. So first one, in total only 1.5% of the human DNA is coding or exon DNA carrying information for proteins or RNA products. Yes. However, when the much larger introns are included in the count, as much as 30% of the human genome consists of genes. That is also correct. So statement 1 is correct. DNA helicase is the enzyme that increase or decrease the extent of DNA underwinding and affects the linking number. Okay. Now DNA helicase, it is an enzyme that unwinds double-stranded DNA by breaking hydrogen bonds between the base pairs, and they are not responsible for any underwinding or overwinding of DNA or changing the linking number. That is the work which is done by the topoisomerases. So statement 2 is incorrect. Statement 1 is correct and your answer is option 3. Then next one. Many species of biennials and perennials are induced to flower by low temperature exposure 0 to 5 degrees Celsius, and the process is called vernalization. That's correct. Vernalization elongates the vegetative period and induces the plant to flower earlier. That is not correct. Vernalization, it shortens the vegetative period and induces flowering earlier. It is not elongating. So statement 1 is correct, statement 2 is false, and your option is option 3. Then next one. Glycolysis involves a series of reactions carried out by a group of soluble enzymes located in both cytosol and plastid. Pentose phosphate pathway is also located both in cytosol and plastids. So both the statements 1 and 2 are correct. Answer is option 1. Porins are not inactivated by temperature up to 90 degrees Celsius. That's correct, because porins are inactivated by temperature up to 134 degrees Celsius. Porin infection is sensitive to radiation treatment. Porin infections are resistant to radiation treatment. So as per me, statement 1 is true but statement 2 is false. But as per your answer key, both statement 1 and 2 are false. So the answer here is option 3. Then next one. Ion exchange chromatography exploits differences in the sign, that is either positive or negative, and magnitude of the net electric charge of proteins at a given pH. So at a given pH, proteins can exist either positively charged or negatively charged, and ion exchange chromatography exploits these differences in the sign and magnitude of the net electric charge of proteins. So that is correct. In ion exchange chromatography, separation can be optimized by gradually changing the pH and or salt concentration. So when you change the pH, there are chances that a negatively charged protein can become positively charged. So based on that, ion exchange chromatography separates the protein molecules. So that is also correct. So both statement 1 and 2 are correct. Chlorophylls A and B are abundant on green plants, and C and D are found in some protist and cyanobacteria. That's correct. Carotenoids are found in all photosynthetic organisms and are integral constituent of thylakoid membrane. Both statements 1 and 2 are correct. Answer is option 1. Auxins have been used commercially for prevention of fruit and leaf drop only. When they mention only, such kind of terms like only, you have to be careful, because sometimes it may be wrong also. Now here the statement is false, because other applications of auxins are also there. Not only prevention of fruit and leaf drop, but they promote root growth from the cuttings, then control stem elongation, cell division, enhances fruit development and retention, and it is also used as herbicide. Then in some plant species, seedless fruits are produced with the help of treatment of unpollinated flowers with auxin. Yes, they induce parthenocarpy. Auxins induce parthenocarpy, producing seedless fruits in some plant species without fertilization. So statement 1 is not correct, but statement 2 is correct, and your answer is option 4. Then next one. During protein synthesis, termination occurs. So terminating signal causing the polymerase to detach from the DNA and release the transcript. That is correct. They have asked for okay. In eukaryotes, polymerase 2 transcribes a sequence AAGUAC. So in eukaryotes, RNA polymerase 2 transcribes DNA into RNA during gene expression, but it doesn't directly participate in protein synthesis termination. So protein synthesis termination occurs during translation, not transcription. And it involves release factors and ribosomes, recognizes stop codons like UAA, UAG, UGA on the mRNA. So that's what happens in eukaryotes. Then in termination, so B is not correct. Termination signal causing the polymerase to undetach from the DNA. No. Termination signal causing the polymerase to detach from the RNA and release the transcript. That's also not correct. Not detach from the RNA, it is DNA. Then in eukaryotes, polymerase 2 transcribes a sequence AUGAA. That's also not correct. So only A is correct. So the answer here is option 3. Which proteins are present in neutrophil? So out of these, collagenase is present, leukotrienes not present, defensins is present, cytokines are not present, alpha 1 antitrypsin is also present. So A, C, E only. Answer is option 3. Molecular techniques used for the identification of microorganisms from environmental samples include metagenomics. Yes, everybody knows that. What is cardfish? Cardfish, it is a technique used to detect specific DNA or RNA sequences within cells. And it is used in gene expression analysis, chromosomal abnormalities detection, cancer research, infectious disease diagnosis like viral RNA detection, etc. Then denaturation gradient gel electrophoresis. So it is used to separate and analyze DNA and RNA molecule based on their melting properties. That is what denaturation gradient gel electrophoresis does. So C is also correct. Then ELISA, no. Polychips. So polychips, they are also known as microarray chips or biochips. They are small solid supports with hundreds to thousands of immobilized probes of DNA or protein or RNA that are arranged in a grid pattern. So that is also used for identification of microorganisms from environmental samples. So A, C, E is the answer. Answer is option 3. Sieve tube elements found in angiosperms are. Sieve tube, yes. P protein present, yes. Sieve plate, yes. Companion cells, yes. So A, B, D, E only. So that is found in option 1. Following are associated with endocrine functioning of pituitary endocrine gland. Pars convoluta, no. Pars intermedia, yes. Pars rhodita, no. Pars nervosa, yes. Pars distalis, yes. E. So B, D, E only. Answer is option 2. Microbes involved in biological fixation of nitrogen are nitrosomonas, yes, involved as Azobacter, Pseudomonas, Anabina, Rhizobium. So all are involved. So A, B, C, D, E are involved. Answer is option 3. Choose the vertebrates from the given list. Dogfish is a vertebrate, silverfish is not a vertebrate, anguilla is a vertebrate, devilfish is a vertebrate, and hippocampus is also a vertebrate. So A, C, D, E, that is found in option 3. Then next one, you have to do match the following. Duncan's muscular dystrophy. Duncan's muscular dystrophy is loss of coordination due to the absence of gene on an X chromosome. So A3, that is found in option 2 only. So your answer is option 2. We will see the others also. Down syndrome is trisomy of chromosome number 21. So short stature resulted by addition of chromosome 21, B1. That's correct. Chronic myelogenous leukemia, like Philadelphia chromosome. Exchange of a large portion of chromosome 22 with a small fragment from the tip of chromosome 9, C4. Then Cry-du-Chat is chromosome 5. Deletion in chromosome 5 resulted in small head. So the answer here is option. Then adenovirus, poliovirus, they have given the viruses and whether they are double-stranded DNA, RNA, single-stranded DNA, RNA, all these things. And the number of nucleotide pairs each are having. Okay, so adenovirus. Adenovirus is double-stranded DNA virus with 35,000 nucleotide pairs. A2. Poliovirus is single-stranded RNA virus with 6,000 nucleotide pairs. Tobacco mosaic virus is single-stranded RNA virus with 7,500 nucleotide pairs. And polyoma virus is double-stranded DNA virus with 4,500 nucleotide pairs. So all that sums up to option 4. A2, B1, C4, D3. Then next one. Use of hypoxanthine thymidine. Okay, in which technique that is used? It is used in hybridoma. A3. 4 can be omitted. 1, 2, 3 we will see. Characterization of F globulin serum proteins, that is by immunoprecipitation. So B1. So it will be option 2, which is more correct. Then formation of chemiluminescent product, that is found in ELISA. Detection and quantification of the number of cells producing cytokines is by ELISpot assay. D2. So A3, B1, C4, D2. Then Markantia. Okay. Bryophyte. The shape of their capsule. So Markantia capsule is oval shaped. A4. A4 is found in option 3. Anthoceros, its capsule is bristle or thorn-like. Polytrichum, capsule is polygonal, and Funaria, capsule is pear-shaped, curved and asymmetrical. D2. So A4, B3, C1, D2. Answer is option 3. Then Platyhelminthes, they are excretory organ. So excretory organ of Platyhelminthes is flame cells. A3. A3 is found in option 3 only. So if you know A itself, your answer is ready. Then B, that is Annelida, it is Nephridia. Arthropoda, it is Malpighian tubules, as you can see in case of cockroaches. And in mollusks, it is feather-like gills. So answer is A3, B2, C1 and D4. Then this question it was omitted because your option doesn't have the correct answer. But anyway, I will tell you the answer. Negatively charged amino acid, it is aspartate. Only aspartate and glutamate are negatively charged. Glutamic acid are negatively charged. So out of this option, A3, polar uncharged amino acid is your cysteine. Aromatic amino acid is phenylalanine and positively charged amino acid is histidine. So A3, B2, C4, D1. Answer is not given in the option. Then next one. Stearic acid. Stearic acid is a non-N-octadecanoic acid. Palmitic acid is N-hexadecanoic acid. Myristic acid is N-tetradecanoic acid and lauric acid is N-dodecanoic acid. So A1, B4, C3, D2, your answer is option 4. So that's it. Thank you for watching my channel. Please do like, share and subscribe. If you want still previous year question papers of life science, you can go to my channel Omni Bio Entrance. Go to playlist section. C for CUCET folder. In that you will find the question papers that are being answered. So thank you for watching my channel. Please do like, share and subscribe. Thank you.